Hello everyone. Today, we begin an important topic, that is general biochemistry, focusing on solutions and the units used to express concentration. This topic is extremely important because almost every biochemical experiment involves the careful preparation and use of solutions for reactions and measurements. Whether in a clinical laboratory, a research lab, or during diagnostic tests, accurate solution preparation is essential for reliable results. So let us understand how solutions are defined, how concentrations are expressed, and why these concepts matter in practical biochemistry. Let us begin with a basic question. What exactly is a solution? We will define it simply and visually, with labeled diagrams and clear examples. In biochemistry, a solution is a homogeneous mixture that contains two main components, a solute and a solvent. A solute is the substance dissolved in a solution. Solutes can be ions, small molecules, or macromolecules like proteins. A solvent is the medium that dissolves the solute. In biochemistry, the most common solvent is water, the universal biological solvent. Examples include salt dissolved in water, glucose in blood plasma, and ethanol in water mixtures used in the lab. Concentration describes how much solute is present relative to the solvent or the total solution value. It controls reaction rates and reproducibility. We will now introduce common concentration units and when to use each: molarity, molality, normality, normality, and percentage solution. Molarity, abbreviated M, is the number of moles of solute per liter of solution. It is the most common concentration unit in biochemistry. The higher the molarity, the more concentrated or strong the solution is. To prepare a 1 molar solution, dissolve 1 mole of solute and make the total volume up to 1 liter with solvent. A molality is the number of moles of solute dissolved in 1 kilogram of solvent. The term molality and molarity should not be confused. While expressing the molality, it is represented by a small m, whereas molarity is represented by an uppercase M. The concentration of a solution could also be expressed in terms of normality. It is based on an alternate chemical unit of mass, called the equivalent weight. The normality of a solution is the concentration expressed as the number of equivalent weights of solute per liter of solution. In a chemical mixture, 1 normal solution contains 1 equivalent weight of solute per liter of solution. The percentage solution could be expressed in terms of weight percent, volume percent, and weight to volume percent, units of solute present in 100 units of solution. In case of preparing a solution based on percentage by weight, one would simply determine what percentage was required and the total quantity to be prepared. Preparation of solutions based on percent by volume, it requires the calculation same as for percent by weight, except that calculations are based on volume. In simple terms, one should plan that what percentage was desired. For example, a 20% by volume aqueous solution of sodium chloride, and the total quantity to be prepared in terms of volume. Volume percent, or volume percent, most often is used when preparing solutions of liquids. This is typically only used for mixtures of liquids. Volume percent is relative to volume of solution, not volume of solvent. The advantage of volume units is that gaseous concentrations reported in these units do not change as a gas is compressed or expanded. Practical tips: always calculate required moles or mass. Choose the correct solvent, and use calibrated volumetric glassware for dilutions. Wear appropriate PPE. For dilutions, use the C1V1 equals C2V2 equation to plan transfers between concentrations and final volumes accurately. Double-check units, perform calculations stepwise, and if possible, prepare stock solutions to minimize repeated measurement errors. Label solutions clearly. Safety note: some solutes are hazardous at high concentrations. Consult safety data sheets and follow laboratory safety protocols at all times. Summary: solutions are homogeneous mixtures of solvent and solvent, and concentrations can be expressed in m% or ppm depending on context. Thanks for watching. practice preparing and converting concentrations to build confidence, and consult protocols for specific biochemical experiments. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.